So here we are at the course webpage, and I'm scrolled down to the bottom, and here's the agenda for day 2. And you can see we are going to start with some introductory slides. And I have, this time I do not have sort of PowerPoint style slides for all the demos. I just have these text and code that we're going to kind of scroll through together, so you can follow along that. You can open these up in your browser and follow along with me and scroll through it with me if you want to. And every one of the lessons also has a worksheet, again, just like last time. This one, the first one, the worksheet here is got blanks to fill in, and then if you get stuck, you can always copy and paste or just run it off of the one with the blanks filled in. Of course, like yesterday, I recommend typing in the blank spaces yourself because it helps you learn. Okay, so let me open up the introductory slides, and I'll go through them real quick. Again, please interrupt with questions or comments anytime you feel moved to do so. So I kind of, as a joke, decided to call this a smorgasbord of options for multi-omics data analysis, because I adapted a lot of the materials for this from the Swedish. The way that grad school works in Europe is, instead of taking semester-long courses, a lot of times students take, they mostly spend their time doing research and take courses, like intensive courses for 2 weeks. You just go and do nothing but take a course and kind of get a semester's worth of credit for this 2 weeks of work. Well, they have one of these courses about omics and bioinformatics analysis that they did, and put all the materials on GitHub so that people could use and modify them, which is really cool. And that's what I did for much of what we're going to do today, because I really am not an expert in this topic. I'm kind of learning along with you, and so it was helpful to have some good teaching materials already made for me to just adapt and not have to write it from scratch. Yes, so there's a picture of a smorgasbord for you. This is intended for you all. I know that many of you work with omics data, whether it be microbiome or gene expression or metabolomics, that kind of thing. And oftentimes you have multiple different omics from the same samples. That's what we call multi-omics, and you might just want more exposure to different methods of analysis, right? And so as I say, I kind of am looking at this at a more big picture level, also because I myself am not a huge expert in the exact nitty gritty of how all these things work, but that's why I call it a smorgasbord, because it's a bunch of options for you to sample from and try a little bit of everything and see what you might find useful. Yes, I'm going to give you a few options because this is just a jumping off point, right? You can't learn all this stuff in one day. I mean, I can't do magic and teach you to be an expert in one day. So I've also compiled some learning resources you might want to use for more in-depth study. Then I'm going to give kind of a basic conceptual overview of some of the methods used for integrating data from different omics together and learning from them. And then we're going to do our code alongs, our code hands-on demos in R. And the demos, I have 5 of them. If we get to them all, I'm not sure, but we're going to do multi-omics factor analysis. That's the first 2 demos. The first one focuses more on kind of the pre-processing and data cleaning steps, which is really important. The second one kind of uses already cleaned data, but focuses more on the downstream steps that you would do to get all the different kind of like graphs you could make and stuff like that. We're going to do one on sparse partial least squares discriminant analysis, which I'll explain what that is soon. A differential expression analysis for RNA-seq data. And the last one is about a technique called Diablo, which is like an extension of the partial least squares when you have multiple omics data. That's what we're going to be looking at. And I know that these are relevant to you because I've seen papers coming out of your research group that use a lot of these techniques, and you know, and you all, at least some people in your research groups or your collaborators, are doing this stuff and using these exact packages that we're going to be using today. So this should be, yeah, this isn't just busy work. This is something that could be very relevant for you. Even if, again, if you don't actually end up using this stuff, it will help you understand the papers that are coming out of your groups and those of your collaborators. So let's go through a few resources for further learning. I just decided to put that first today. Well, I just wanted to point you to this very useful short course that the American Stat Association put together on statistics and genomics and genetics, which covers a lot of the topics in more depth that we're going to be talking about. It's got some lectures on YouTube. Again, so if you go to the course website and you click on the slides, you can go to all these links directly from the slides. So you have access to these links. MultiOmics is also one that we are going to be using. There's an R package called MultiOmics, but it's also kind of like this research collaboration of people that have put together a whole lot of training materials on how to do this kind of analysis. So it's really like kind of a fully integrated pipeline, and it teaches you how to use it every step of the way. There's like ebooks, self-guided courses, sort of like YouTube videos, and they have online courses that they offer every few months that go for a few weeks and are kind of like asynchronous ones, which are pretty cool. You should definitely take a look. And as I already mentioned, the sort of state-funded bioinformatics research agency in Sweden is the one that has sponsored this short course that for graduate students in bioinformatics. Unfortunately, folks from the United States can't participate directly in the course, but you can look at all their course notes and materials and everything. And so that's what a lot of what we're going to be doing today is based on. Okay, so the next part of this presentation is about the conceptual framework, or how some of these omics analyses work, that we're going to be doing hands-on later today. So when we're talking about multi-omics data, what exactly are we talking about? How are those data structured? Well, it's anytime that we have more than one omics data set from the same sample. And an omics data set is basically a multivariate data set with many different columns, basically representing different chemical compounds, like or different genes. Like the genome is obviously a type of omic data. Methylome, looking at sites on chromosomes, the methylation pattern of the genome is called the methylome. The metabolome, all the different compounds that are present in a sample. Transcriptome, that would be all the RNA that's expressed in a sample, and microbiome, the taxa of microorganisms present in a sample, right? So anytime when you have more than one of those from the same biological unit, an organism or a cell or a sample. And yes, so these can be it depends on what biological unit the omics come from. Many come from organism, like the gut microbiome, for example, but there's even single cell omics. Like you could look at RNA transcriptome from individual cells within an organism, but is also commonly done. And usually when you have omics data, you also have some sort of metadata or covariates that you want to associate with those omics patterns. Like you all do a lot of experiments where you have treatments, you have you subject people or animals to different interventions and then look at what effect that treatment has on, let's say, their gene expression or the microbiome or whatever. And then you also have other covariates like age, sex, whatever clinical things you measure on your individuals. You have also outcome variables, like if you're concerned with the presence or absence of some disease or whether the organism died or whatever. Those are things you could try to correlate with your omics data. And there you could also look at the cell level, right? If you're doing single cell omics, you might have cells from different tissues and compare the gene expression between them, for example. Those are different covariates you might have. Sorry, are there any questions so far? Yeah, this is pretty basic stuff so far. So what can we use these kind of data sets for? What questions can we answer with them? Well, first, before we think about the outcomes and everything, we want to look at just relationships between the omics data themselves. Like, are certain expression of certain genes high or low associated with the production of certain metabolites or the abundance of certain taxonomic groups in the microbiome? We could look at that. And then going beyond kind of within omics, we could then relate our omics data to the outcomes. Like, can we predict whether someone is going to get a disease based on gene expression in their cells or based on what bacteria are living inside of them, the microbiome and that kind of thing? And can we get an even better prediction if we combine multiple omics data sets together to predict that outcome? Those are some really cool questions we can answer with multi-omics data. So luckily, we talked about machine learning yesterday, right? And talked about supervised and unsupervised learning, because that is also relevant here. There are supervised and unsupervised techniques for omics analysis. So you can see we're building on our knowledge from yesterday. Right, so in the context of the omics data, supervised would be when we have some sort of outcome variable that we know what happened to each individual. For example, oftentimes it's like a disease status, and we want to figure out what combinations of features, like what metabolites or what taxa in the microbiome combined in those different omics data sets will predict that outcome that we know about. We know what groups the individuals belong to. We say, okay, you all got the disease and you all didn't. But unsupervised, there are also unsupervised techniques for omics analysis where we don't have an outcome. We're just exploring relationships between the data sets, right? Like like I said before, is there certain microbiome taxa that are associated with certain aspects of the metabolome and that kind of thing. So focusing on supervised methods, the ones where we know what groups everyone belongs to, there's kind of two groups within that: descriptive and predictive. And descriptive means we want to go into our omics data sets and find weights, like sort of estimate parameters for each of the variables in the omics data set that will optimally separate the classes. Like if we, which ones of those variables explain the most variation in between the groups and can be used to separate them. And there are different sort of techniques we can use to estimate those parameters and determine what is optimal separation of the classes. Now, predictive supervised omics analysis would mean if we take, this is like the machine learning stuff we're talking about yesterday. If we have a new sample that we only know its omics, but we don't know its outcome, can we predict its outcome, like whether it got the disease from its microbiome, metabolome, and so forth? And so they call that constructing a rule or a classifier that will take the input of the omics and predict the output of whether it got the disease or what group it belongs to. And again, with prediction, it's a lot easier to say whether you did a good job or not, right? Because you can have validation data and say, well, are you right or wrong? How many false positives and false negatives did you get? That's called the sensitivity and the specificity, respectively. And there's like different metrics for that. So it's like with prediction, it's almost easier because you have this very tangible way of knowing if you were right or wrong. Descriptive is a little more fuzzy, right? Now, what kind of method we use depends on the size of the omics data. Omics data come in all shapes and sizes. Usually, and kind of what size it is determined by what kind of omics data it is. Usually, not always, but usually, metabolome is smaller in terms of how many different variables it has. Like it's very expensive to generate metabolome data for thousands of compounds. So usually metabolomics data tend to be on the order of tens to hundreds of compounds of metabolites. Those are the features, the predictor variables. Then next order of magnitude would be your microbiome and your transcriptome data sets. Those tend to be kind of bigger. They tend to have on the order of thousands of variables. But the really big ones would be genomic and methylome data. Those are like all the SNPs in the genome, or all the locations that are either methylated or not. There can be many thousands, hundreds of thousands of features in those. So those are huge data sets, right? But that's only how many variables are in the data set. Well, also it depends how many samples you have. So we were talking, those are called P and N, right? The one we just talked about, the number of predictor variables is called P. Again, that can vary a lot, right? Like I said, it could be a couple dozen metabolites or hundreds of thousands in your genome. But N is the number of samples. Now, it does not vary quite as much, right? Like we're talking about clinical biomedical research here. You're going to have, somewhere like 10 to 100 patients or animals. Like maybe there will be some huge study where you'd have 1,000, but that would be like an incredibly big study. It doesn't vary as much. Like it tends to be in the small to medium range. So then we have very different dimensionality of the data set depending on this sort of the ratio between P and N. If you have a data set with relatively few predictor variables, relatively few features, and less smaller P than N, you can use Bayesian methods, which are kind of, I would say, the best, but they're very computationally intensive and do not scale well to having tons and tons and tons of variables. So they're the most useful with data sets that are kind of not as wide. They don't have as many variables in them. Then kind of when you get to having a little bit more predictor variables, then you can switch to kind of your conventional statistics, your frequentist methods. But when you have really wide data, you have a lot more variables than you have samples, P is much greater than N, you can then go and use kind of deep learning or more machine learning methods, which have really good predictive performance on big data, but don't work as well with smaller data. And so your technique that you use depends on the size of your data and the shape of your data. Any questions at all? And software, what can we use? Well, R has a lot of really good packages for this kind of analysis, and there's kind of, you all might be familiar with this. Yesterday I talked about CRAN is the main platform where people download R packages from. there's kind of a second one called bioconductor, which specializes on bioinformatics packages, and that's where a lot of these omics packages are hosted. Python also has a lot of omics packages as well, and actually, one of the analyses we're going to do, even though we're going to do it within R, is going to be running Python behind the scenes to actually do the actual algorithm is coded in Python. So many people do this type of stuff in Python as well. And there's also kind of an online platform called MetaboAnalyst, which is focused more on metabolome data, but can be used for other omics data. And it kind of has like a point and click, select options from different menus, and it can be easier for beginners. And the nice thing about it is it is actually running R behind the scenes, so you can export an R script from it and see what it did in R and modify it if you need to. So that's also a nice option, especially when you're just getting started with this stuff. Okay, we'll go through a few more slides about the process before we get in the hands-on stuff. This is an interesting figure that people often cite this statistic that data scientists spend more than half their time cleaning and organizing the data, sometimes collecting the data sets, and only this, 20% or so of the time is actually spent making models with the data, right? It's like data cleaning is the most time-consuming part. So it's an important part. If that's what you spend the most time on, it behooves you to do a good job of it. Well, yeah, we talked about this a little bit yesterday in the machine learning thing. Some of this stuff is the same here. It's important to look at the features, the predictor variables. If some of them are all 0 or all the same value or almost all the same value, they can be removed before analysis because they carry little to no information, and all they're going to do is bog down your analysis and make it use up more memory and time for no benefit. So it's good to remove those beforehand. Yes, Vara.
So I have a question. So to remove the certain features, right? Like, is there any criteria like we have to filter, right? Like, or it depends upon the literature.
Yeah. So yes, there is a little bit of subjectivity in what you would use as the cutoff. I mean, obviously, if one is all zeros, you can remove that, right? You might as well. But what if it's like 2 out of 100 zero are not 0, or 3 out of 100 or something like that? Well, I think there's a little subjectivity. As long as you could look in what the literature does, what is a common one, or you could try different thresholds and look at whether your results are sensitive to the choice of threshold. And if they're relatively insensitive, then it doesn't matter as much, right? But if your results change a lot by this small tweak to where you take your 0 cutoff, that's kind of a bad sign that your patterns are not very robust to those small changes, right? Yeah, so that would be like removing the columns, the predictor variables with no variance. But what about removing rows, like removing entire individuals or samples from the data set? You might need to if you have too high of a proportion. There's often these omics data sets have a lot of missing values in them, individuals for which we could only get a subset of the metabolites or the genes or whatever. Many of these methods can handle it if some individuals have missing data. It would be a bummer to throw out an entire individual just because it had, a few out of 1,000 loci were missing for that individual. But that only works up to a point. If, the majority of values for an individual are missing, it's not holding that much information either. So those may be good to remove as well. Again, there's kind of a gray area of what percent missing values is the good cutoff to throw out, And, but you don't always have to throw them out. Again, like I said, if there's only a moderate number of missing values, there are a lot of methods to impute the missing values, to kind of use a separate statistical model to predict what those missing values might be before we feed that into our real main model. Keeping in mind that those are just our best guess, right? And there's some uncertainty associated with those imputations, but you don't want to have to throw out too many of your data points. If you have only moderate missing values, I would recommend doing some imputation. I'm not going to talk about that much today. I just wanted to raise that point. Okay, so exploratory data analysis. The first thing that you often want to do when you have omics data is to do kind of I would say, granddaddy of all multivariate analysis techniques, which is like a principal components analysis. It is the simplest type, I would say, of multivariate data analysis. It was invented in 1901. Like my great grandparents were not even born yet when PCA was invented. I mean, it stood the test of time. Like it's still a great method today to basically take a very high dimensional data set and reduce it to a smaller number of dimensions that have as much information as possible to reconstruct that huge many dimension data set from a smaller amount of dimensions. So we can actually compress down the information in a data set with fewer and give the same amount of information with fewer numbers. Like the same way that in computer science, you would take an image and then compress it into a JPEG that basically takes up less space on your computer, but looks just like the original picture. You only lose a little bit of the information in the image, and it takes up much less, megabytes to store that image because it's doing something basically like a PCA on the picture. So yeah, you start with a small n and big P, right? We have a lot of variables, like a lot of genes and a moderate number of individuals, and it turns it into a smaller number of components and a product of components and loading vectors. So each like this would be an example with trying to reduce this from P variables to only 2 components that have as much of the information in the original data set as possible. And then so if you start with an N by P matrix, and then you end up with an N by 2 matrix and a 2 by P matrix that you then multiply together and see how much of the information in the data is preserved. So every sample has a component score, and each one of the N samples has a component score, and each one of the P features has a loading vector. That's like a lot of the methods that we're going to be doing, even though they're more complicated than PCA, are basically doing that. They're reducing the dimensionality of a very, very complex data set into a smaller number of dimensions that have as much information about the original data set in them as possible. That's kind of what we're doing every time. PCA is really quick to run. It's not computationally intensive, and it will, in many cases, kind of tell you what you need to know. Especially, it'll give you a quick idea of the structure of the data. And it can also be used as kind of a diagnostic tool since it's so quick to run, which will help you see if there's any errors in the data, if anything looks weird, if there's any biases, like if something sorts out in a way that you didn't expect. It could be some sampling error or something like that you can then notice real quick before you get too in the weeds with complicated omics analysis. So that's a great first step every time, I think. Now, the last few slides here before we get started, I'm just going to go through and introduce you one by one to the different techniques we're going to be going through today in our hands-on demos before we actually get started. So I should update this slide. This only has some of the methods we're going to be doing today, but yeah, the multi-omics factor analysis is an unsupervised omics analysis. We're looking at relationships within the omics, not associating them with some particular outcome, at least at first. And then SPLSDA is a supervised one where we actually are using information about what groups the samples belong to in the analysis to try to predict what groups new samples belong to. A note to update that slide. And there are a lot of other methods out there that we just don't have time, or I don't have the expertise to cover. Like, there's a lot of sort of machine learning based methods that are kind of like, I would say, PCA on steroids, like a more performant version of PCA that scales better to really, really, really big data sets like we have in some of the omics data. And yeah, we're just not going to have time to cover those, but you see them a lot in the literature. Deep learning and neural networks obviously are a big thing and used a lot for omics data. And there's like an entire school of thought based on network analysis, sort of looking at kind of graphs of connections between nodes, which are represented by the different samples in your data set. Again, they're really useful, but we just can't cover everything. But those are ones to also look out for. And here, I just wanted to just give a graphical example of some of these machine learning dimension reduction methods that are used, that you might see these terms in papers with omics data, TSNE and UMAP. Those are some of the names of these machine learning analogs of PCA. Like I said, it's kind of like a PCA on steroids. And they can be used to create a consensus mapping, basically take multiple omics data sets and do a clustering of them and see what groups form in the data. Like this just shows how, if you have data that are obviously, there's just like different points from different continents. Right, these are colored by what continent they belong to, although the machine learning model does not know what continent the points belong to. You can see this is PCA results. It does a pretty good job separating them, as well as and the different machine learning techniques also like can tell what they can sort of find natural groupings in the data, and those are like the continents. That's just an example of how these results look. So yeah, they're kind of comparable to PCA, but perform better on really big data sets. And the deep learning methods, as I said, are pretty cool. They actually, on very big data sets, are the best. They're the cream of the crop. They do the best at predicting of any model that we've ever been able to come up with. But really, they're not like that different at their heart than many of these other methods. It's a dimension reduction, right? Like taking a complicated an n by p data set and reducing it and explaining as much variation as possible with fewer variables, and then do a clustering. The algorithms are more complicated, but the basic idea of what it's doing is similar to all the other things. And another thing that's good about these type of deep learning methods is that they're very good at picking out nonlinear patterns as well. Different thresholds or funny shaped relationships between variables. And a lot of times what people do is they'll fit a deep learning model to an omics data set, and then they'll, in the second step, stick the results of that into one of these like machine learning visualization methods, like I just talked about, to help mainly just to make a cool graph showing the results. It's kind of a two-step approach. Network analysis, as I mentioned, is a big one. Basically, what you do is take your omics data set and generate a network graph showing which nodes, which samples are connected to each other based on what variables they share. And you can do a network for each of your omics data sets. And then there's a method called similarity network fusion that will combine sort of take the overlap of the networks from each of your omics into a single fused network, which can then show overall relationships between your samples, basically. Pretty cool techniques, but we are not going to cover them today. And the network then, so then that overall network can be used to identify which features, which genes or metabolites or whatever are most important for explaining the relationships between your samples. Okay, so here are the ones we actually are going to talk about today. First and foremost, we're going to talk about, first we're going to talk about multi-omics factor analysis. It is a data exploration methodology, right? It's an unsupervised technique. We do not have a hypothesis about how the groups are, that we're not hypothesizing this group of samples is different mean than that group of samples. We're just looking at relationships in the data here. And it can take multiple omics layers like metabolome, genome, proteome, microbiome, and extract common axes of variation from them. So across all these omics, are there certain variables that are associated with one another within samples? And again, just like a lot of these methods, it's taking data sets with a lot of variables and using them to construct this low dimensional representation that contains as much information as possible about the original data set with fewer numbers. And those are called the hidden factors. We reduce our many variable data set. We have many loci in the genome and many metabolites, and reduce that to a moderate number of factors. And a nice thing about this is it can handle missing values, and it can be used to impute missing values in the data set. So it can actually take, not only can it handle data with missing values, it can actually make predictions because it's a generative model. It can predict the missing values for the samples that had missing values in them, which is kind of a nice touch. And yeah, again, it's nice for making visualizations, and also it can help you come up with biological interpretations for some of these factors. And we'll go through this in the example. That it will make more sense when we do. Oh, by the way, I forgot to mention this before. If some of you were in this, I did this workshop in Little Rock last January. Some of this we did last year. So if this looks familiar to you, it's because we did do some of this stuff last year, but I added some new content as well. So there'll be some old and some new if you were here last year. I think a few of you were. The next one we're going to do is the partial least squares discriminant analysis. And again, it is supervised method. We don't just have our N by P matrix of samples by features in our omics. We also have a Y variable, an outcome, like what group does each sample belong to. So, and we basically choose a number of groups to kind of reduce the dimensionality of our data set to, and we want to come up with the groups are our outcome variable, and we want to find a set of components, a lower dimensional representation of the data that not only explains the variation in the omics data, but also maximizes the variability between those groups and minimizes the variability within groups. So just like kind of a regression analysis would do. So yeah, then that can be used to predict the group a new sample would belong to. Now, that was partial least squares discriminant analysis. What we're going to do is a special variant of that called sparse partial least squares discriminant analysis. It is there's kind of a technique called sparse PCA. So it takes that and combines it with a partial least squares analysis, where basically it does variable selection, which turns some of the coefficients for each of the components to 0. You see here how some of these loading vectors contain 0. It uses a technique similar to lasso regression. hey, didn't we see that yesterday when we were talking about machine learning? Yes, we did. It's using a regularization parameter, as we learned about in our machine learning workshop yesterday, to sort of shrink away some of those coefficients, avoid overfitting the data, so that we're not fitting the noise in our data set, and we're better at predicting on new samples. Yeah, so it's kind of a machine learning approach. And just like we did yesterday in machine learning, the way you determine the optimal amount of shrinkage, the optimal amount of regularization, is cross-validation. And hopefully you all remember from yesterday how that works. We fit the model to part of the data and predict the other part. Then we keep switching out which part was held out until we've predicted all of the data one time. That's cross-validation. And that is used to train the model to estimate the best parameters. And then just like we did yesterday, we take an independent test set and predict the groups for it, so we can see how well we did. There's also a separate cross-validation step that can be done to assess the performance of the model on the test set. Last couple of slides. Another thing one of our last demos we're going to be doing today is called Diablo. That stands for Data Integration Analysis for Biomarker Discovery Using Latent Variable Approaches for Omics Studies. Kind of a mouthful, but basically all it is a kind of a special variant of that sparse partial least squares that we just talked about, that allows you to integrate multiple omics data sets together. So it's basically the same technique we just talked about, but SPLSDA is only for one omics data set, and Diablo is basically just doing it with more than one together. So it's kind of useful. And lastly, I also wanted to just talk a little bit about differential expression analysis and do a demo for it, since all of these ones I've talked about so far are like multivariate analyses, looking at variation across an entire data set. But there's also kind of a simpler way to do it, which can be useful, which is just to take each variable in your data set individually and just do a statistical model on it. And like if you have treatments, and then you say, okay, does our treatment cause this gene to have higher or lower expression, right? Is it upregulated or downregulated? Then look at the next gene. Does the treatment is there a difference between the treatments for that gene? Do it for every gene. There's nothing wrong with doing it that way. And basically you're just doing a separate statistical model for every gene in your RNA-seq data set, and then do a correction for multiple comparisons. That's kind of like the old school way of dealing with multivariate data, and it's still a good way. So I felt it was a good one to talk, because you all also use it in your papers as well. And if you've ever seen the DE-seq2 package in R, that's the one where it's implemented. That's what we're going to use today. Yes, Bharat.
I have a question. So in the previous, the partial least square, right? Like there uses the samples and also different futures, right? So the futures means it's also single metabolite, right?
Yeah, each feature is a metabolite, yes.
Right. So what's the difference then, this differential gene expression analysis and that too, because it is also single.
So this differential expression analysis is basically looking at each feature in isolation. It is not looking at patterns of variation across the entire data set. It's just doing basically almost like a t-test for every gene or every metabolite separately. The other omics analyses techniques that I've talked about are not doing that. They are looking at relationships across it's sort of a more holistic way of looking at it, looking at relationships across all the metabolites and seeing if they're correlated with each other. It's like looking at not only at the relationship between the metabolite and the treatments, but also the relationship of the metabolites with each other as well. But this DE-seq does not do that. It's just like counting up which ones have more and which ones have less based on the treatments. Yet then you do a correction of the p-values because you have multiple comparison. But other than that, is it's looking at them each in isolation. So that's the difference there. Does that make sense?
Yes. Yes. Thank you.
Okay, cool. Well, that is all the introductory slides I have. So why don't we can just take a quick 5-minute break for folks to step get up and stretch if they need to. And so let's come back at yeah, was that 10:24 Central Time, and we'll start on the hands-on part. I will be right back. Hey, before we get started again, I see we have a new person on that wasn't on yesterday. Yi Hong, do you need help getting set up with anything? Do you have like R installed and all that stuff? Just let me know if you do need help with any of that. We can help you get set up so that you can follow along with everything. Well, anyway, let me know if or anyone gets stuck. Let me know because our TA Grant is he'll be back later, but he's not gonna be on for the next half hour. So, but I'm happy to pause things and help you out if you need help before he gets back. But after Grant gets back, he will make a separate Zoom room for anybody who needs help with anything technical. He can help you. But that'll be at about in half an hour. He'll be back. Okay. I think yes, Megan.
Just a quick question. I know you mentioned it yesterday, but where are the homework assignments?
Yeah, well, I didn't do exercises for the ones for today, but for yesterday, all these lessons have exercises. If you just go to if you just like click on the text version of the lesson
Okay.
and scroll all the way to the bottom, there's some little exercises there. Yep, just down here at
Okay.
the bottom, and then there's a separate page with the answers on it. It's
Okay,
if you
perfect.
want to do those, yeah. Okay, so let's go ahead and everybody go back to the workshop page, and for each of these demos we're going to do, we're going to download the worksheet for each one. Let's just go ahead and download them all now and save them to a folder so that you will have them and open them as we go. So I'm going to go ahead and do that on mine, and you can do it as well. So I'm going to click on this worksheet for demo 1. I'll save it in a folder.
I have a quick question.
Yes, please.
Where do I find the MultiOmics package? Because mine isn't like already installed, and I did the big installation with like all the packages, and I didn't see it in there.
Okay. It should be. I'll pull up that script in just a second and walk through installing that. Just a second.
Okay, thank you.
Yep. So let's see. We'll go ahead and I'm just going to save all the worksheets for these 5 demos in the same folder. There's number 3, 4, and number 5. Okay, so yeah, Riley, if you go back up to the top of the workshop page and this setup script here that's in the pre-workshop instructions, that has the script for installing all the packages that we're going to need. So if you, yes, I'll save that as well. I'll open that and show you the installation for it. I think the MultiOmics package should be on there. So let's go into R. I'll open up the setup script. Yep. So basically, first we installed all the packages that are on the main R package repository, and then you have to use this BIOC manager installation to install the packages that are on Bioconductor. Those are on the special bioinformatics package repository. So you have to run this line as well, and that'll install MultiOmics along with the other packages we're going to be using today. So yeah, this is what you have to run. Got that?
Yes, thank you.
Cool. Okay, so I'm going to also open up the worksheet for today's lesson, or for the first demo. Here it is. I'm going to open it. Okay. Yes. Okay. Well, that shouldn't be on there anymore. I'll explain that as we go. Okay, so I'm going to basically switch back and forth between going through the text of the lesson and then running code in R. And I'll explain kind of what we're doing as we go. So I'm going to go ahead and open the lesson text for this lesson demo number 1. We're doing a case study of single-cell omics data from a mouse study with the multi-omics factor analysis. So yeah, so basically I am not the original author of these materials, but I've modified them a bit for our use. Okay, so basically, yeah, let me make this a little bigger so you all can see this, but you can also open this file on your own browser and go through it. Hopefully that's big enough for people to look at. Basically, we're going to use this technique, multi-omics factor analysis, to look at multiple single-cell omics data sets taken from different, basically stem cell types within a mouse and see if there are patterns that are common across the omics from those different stem cell types in the mouse. And we have 3 different omics taken from each of the cells. And so I'll talk to you about each of them as we go. Okay, so basically we have 3 omics, like I said. The chromatin accessibility omics data set is called ATAC. The methylation methylome data done by bisulfite techniques, that's called BS, and we have gene expression. We have RNA-seq information from the same cells, and there are 2 types of cells that they sampled. One are called embryonic stem cells, and the other is called embryonic bodies. But because this is an unsupervised analysis, we do not tell the model basically which group each of these each sample belongs to. The model doesn't know it. The model is just looking for patterns in the data, and then we can later say, oh, these patterns kind of correspond to those two different cell types. Okay, so we're going to first off load the R packages, which hopefully everyone has installed, using the library calls at the top of our script. So I'll switch back to RStudio, and the 2 packages we're going to load are MultiOmics. Should have a capital O, and this is also where you can see if your installations work properly. Otherwise, these won't load. And MOFA2. Those are the 2 packages that you should hopefully have installed and ready to go. So I'll highlight that and hit Control Enter. Okay, so has everybody got those packages installed and can load them with the library call in RStudio? Please let me know if not, since we might as well make sure everyone's on the same page and not have anyone be left behind. And now this line here, you should not run. Skip that line, because I figured out a better way to get everything to run that does not require that, but I forgot to remove it from the worksheet yesterday when I was updating everything. Okay. Yeah, but please speak up or put in the chat if you have any problems right now, since Grant is not here at the moment to help out. Okay, so now we're going to read in each of the 3 omics data sets, do a little cleaning up, take a look at them, and do a little bit of data, I would say, cleaning or data processing on them so that they'll work for our model. First, we're going to read in the single cell RNA-seq data and a lookup table that has more informative names for the genes in this RNA-seq data set. Okay, so we're going to I included code here. This one lines 9 and 10 will not work because you're not on the demo server. You're running this off your own machines. So you should run lines 13 and 14, which will import the data from a web location where I've posted the files. So go ahead and run those 2 lines there. So that should take a second to download those and import them. Okay. Going back to the lesson. Okay, what we're going to do is we're going to use this lookup table to match the ID. So the data set only has numeric IDs for each of the genes, but we can connect them to gene names because those might actually be informative to us. And so this first line of code basically sorts the lookup tables so that it's in the same order as the data. And then we're going to turn the column names of the RNA-seq data set to this external gene name column from the lookup table. Then we're going to transpose the data and then just print the first 5 rows and the first 5 columns of it. Right? We learned yesterday how to subset data frames by rows and columns. That's what we're going to do. So let's go ahead and do that. So I've left you some blanks here on line 19. Call names. That means we're going to set the column names of our RNA-seq data set. And we're going to set them from the column called external gene name from our lookup table. Let's run these 4 lines of code. And you can see what does our data set look like. Let's take a look. Well, we have each of these, the rows now are since we transposed the data, the rows are now the individual genes. So now we have names for them. MRPL15 is one of the genes, and the columns are the individual samples. So like ESC was for embryonic stem cell. So this is like embryonic stem cell number A2. That's one of our cells that we got RNA-seq data from. And so it's basically how much RNA transcript there was in that cell of each of these many genes. So like it's a continuous variable, right? Like this cell had 500 and some whatever units of transcript of this gene, it had 0 of another, right? So it's got to be either 0 or a positive number, but it can be a continuous variable. It can go, there's no sort of upper limit there. Everybody good so far? Is everyone able to follow along? Cool. Now, again, we have 2 other omics data sets, the single cell bisulfite sequencing data, which is the methylome, and then the chromatin accessibility omics data set. Again, we're going to read these from the web location since we're not on the demo server. And basically, the next few lines of code, we transpose those data sets as well so that the because apparently for the MOFA analysis, we need the rows to be the individual variables or genes and the columns to be the samples. So we're gonna, T is the transpose function. It'll swap the rows and columns of a matrix. So that's what that's doing. And then we're just gonna look at the first rows and columns, the first five by five from the corner of each of the data sets to see what they look like. So we're gonna import using lines 29 and 30, run that to import those two other data sets from the location on the web where I posted them. Okay, and let's look at the bisulfite one, run these two lines, the methylome one. Okay, you can see that here the columns have the same name as the RNA-seq data set, right? So this is the same cell, ESC number A2. But these are different row names because these are different variables. This is a whole different data set. But the methylation is actually different structure than the RNA-seq. It is actually a percentage between 0 and 100. The bisulfite sequencing method detects methylation as a continuous variable. Like 0, it's completely unmethylated, and 100 is completely methylated. So many of the numbers are either 0 or 100, but you can see there's also, 30 and 90. There's some that are in between as well. And let's look at the chromatin accessibility as well by running the next two lines. That has the same format. Again, the column names are the same cells again, but we also have between 0 and 100, but it can be in between, like 0 completely inaccessible, 100 completely accessible, and in between as well. So far, so good.
Excuse me, I didn't get the second data set.
Didn't get what?
got
I'm sorry.
the second data set. So you got it by running line 29 and 30?
That's right. It didn't work?
No,
Did you get an error?
I did. It's saying file RT. Can I open connection?
And are you are you connected to the internet? Did you get an old version of the worksheet, or did you download the one just now?
I download the one just now. I download the fill-in one.
Okay, yeah. Try it one more time. Do you get the same error?
Well, yes.
That's weird. Same error?
It look like it's trying to work, but it's not. Like it just comes up like it's not an error. It's just saying that it's reading the limited file.
I see. It's just like being slow or what?
Well, the stop sign is gone.
The stop, but did the error pop up? Do you have like in your environment? Do you see one each of these three things in there?
Yes.
You see them now?
I see those, but I don't see the table like you have in the
okay, well, yeah, then you have to run these following lines for that. Good. So it seems like maybe it just didn't download the first time or something.
Okay.
I don't know, like if the connection timed out or whatever, it might have to try it twice or something. Okay, so so you're all good?
I try to run it.
Let me know if you're not good.
So I'm not getting the table, but you saying okay, there it is. It's just slow. Okay, I got it.
All good?
Yes.
Oh, yeah, sometimes when I'm trying to do this, like when I'm on Teams, it uses a lot of the processor, and then like everything runs slowly.
Thank you.
Okay, cool. Yeah, you're welcome. Okay, so basically, here's some of the data cleaning or data pre-processing steps we might need to do if we're working with omics data. Okay, we want, first of all, we're going to do two different things to the RNA-seq data set. We're going to get rid of features of genes that have low expression overall. We only want genes that have like a decently high mean expression. Again, I am not the expert on this, so I don't know how this person came up with this threshold, but they want genes that have a mean of at least 1 to keep. So we're going to get rid of any rows that have keep only rows that have a mean greater than or equal to 1. That's what it says, row means greater than or equal to 1. And we're also going to do a log transformation on the data set because we want to look at variation in expression on a log scale because it varies by orders of magnitude, and it's these multiplicative differences in expression that matter. So that's why it kind of also makes the data behave better because it has more normal distribution at that point, which can work better for some of these analyses. So that's what these two things are doing. First, we're getting rid of any gene that has a mean of less than 1. And then we're gonna take the log of it plus 1, right? Because we have a lot of zeros, so we have to add 1 before we take the log. And then we're going to look at the data again after that. That's what we're doing for the RNA-seq. You can see right now, RNA-seq has 12,314 rows. Let's see how many are left after we do this little filtering step. We're going to use the row means function to get the mean of each row. And we want to keep only the ones that are greater than or equal to 1. Okay. And then we're going to use, in the next line, log 10 function to take the logarithm base 10 of the gene expression values. So I'm going to hit Control Enter to run line 42, and Control Enter to run line 43. Now I'm going to run line 44, which is going to look at the first few rows and columns of the data. You can see now that instead of going up to 500 and whatever, it goes up to 2.7 there, right? Because that's like the log base 10 of 500. And then we're going to look at the dimensions as well using the dim function. You can see we only got rid of a few. It was not a very severe filtering. We still have over 12. We had 12,300 genes. Now we have 12,100. So it only got rid of a very few genes. Okay, now what are we going to do with the other two data sets, right? We have the methylation and the chromatin accessibility data sets. Well, like I said, those vary from 0 to 100, and there's some in between. But really, a site can either be methylated or unmethylated. It really should be mostly 0 and 100. There shouldn't be too many between. So what we want to do is, first of all, we're going to reduce the dimensions of our data set by getting rid of some of the features in each of those data sets that don't have very high variance. Like if almost all samples have 0 for a particular locus, or if all samples have 100 for a particular locus, it's carrying no variation. It's not going to give us any information when we do our omics analysis. We can get rid of it. Or there's also this function near 0 var from the MultiOmics package gets rid of not only ones that are all the same, but ones that have only a very low variability as well. So we're going to do that for the methylation, the bisulfite sequencing data set. with a near zero var function. So we're going to call this near zero var is what we're going to use. I'm going to hit Control Enter to run that line. And to take a look at it, we're going to look at the first few rows of the metrics with a capital M object from inside of that near zero variation object we just created. We're going to look at that. So you can see it has, this is telling us which of the loci have only a very small percentage of unique values in them. So we probably, they have low variation and we can filter them out without losing much information. And in line 51, we can call dim on that as well to see how many we're getting rid of. All right, so our bisulfite sequence data set has, if you look, it has 8,500 rows in it right now. And our filtering step has identified about 3,000 loci that we can say have little variation and we can filter out. So we're actually getting rid of a pretty decent fraction of our loci in this way. And then this step here is being used to filter the bisulfite data set, getting rid of any rows in which that match the rows in the ones that were identified as having low variability. So it's saying there's why this minus is here. So it's saying get rid of any rows in our data set whose names match the rows in the ones that have low variability. That's what this line is doing. And then we're going to look at the top, at the first few rows and columns there as well. So the row names is that function there. And then, yes, metrics is the name of the object that has the row names for the ones that we're going to get rid of. Metrics. Run that line. Look at the first few rows and columns of the bisulfite data set now. You know, it doesn't look too different, but we got rid of some of the rows. So what are the dimensions? Yes, now we're down to about 5,000. Remember, we had 8,000 to begin with, got rid of 3,000 with low variability. Yes, Jasmine, question.
Can you hear me?
Yeah, I can hear you.
For some reason, when I did my thing that you have there, I got 0 rows, and then when I look at the 5 by thing, I have all NA.
Okay, so yes, probably there's a typo in your code somewhere, I would say. So
Okay.
I could try to find it, but it might be easier for you to just go and copy and paste off the worksheet with everything filled in.
Sounds good. Thank you.
Although, but you'll need to go back and reload the original SCBS data set from the URL again, because you've now kind of like deleted all the data in it. So you'll have to go back and re-import it and run it from the top.
Run it from the top. Okay, sounds
Yep.
good. Thank you.
Yes, sorry about that. Okay, and now we're going to do the same thing for the third omics data set, the chromatin accessibility data set, except we're going to use this unique cut equals 1. We're going to use a slightly less severe filtering and only get rid of things that have less than 1% unique values in them. And so that's what we're going to do for this one. I've left you no blanks to fill in this part. So I'll just run each line one at a time by hitting Control Enter each time so we can see what it's doing. So we'll do that for this this data set. We'll take a look. You can see that it has gotten rid of or Those are the ones that have less than 1% unique. We're going to look at the dimensions of how many it's getting rid of. It's 91 only. So you can see that our chromatin accessibility data set has 11,000 rows in it. It's only going to get rid of 91 here. And then this line 59 is the one that's actually filtering it out. Let's look at the first few rows. Okay, still looks good, right? Is just a good diagnostic to check we didn't accidentally delete all the data. And then how many, what is the dimension of the data we have left? We have, right, we got rid of about 100. We had 11,800, now we have 11,700. So far, so good. Okay. Now, let's take a look at the distribution of the data in each of the omics that we have, because our statistical model that we're going to do requires us to have an idea of how the data are distributed. Then we can determine what statistical distribution we should use to model them. So the histogram can help us look at that. So we're going to look at the row mean, so like the average expression for each gene across all the samples for this RNA-seq data set as a histogram. So on line 65, we're going to use the hist function to plot a histogram. The data we're going to use are the row means of the RNA-seq data. And we're going to use breaks equals 100 to make a histogram with 100 bars on it. And that's going to be the RNA-seq data set. The main means what title the plot will have. I'm going to hit Control-Enter to run that. So you can see that the RNA-seq data are kind of like more or less continuous. We have a decent number of low ones and kind of a tail of highly expressed genes. And again, this is the log transformation that we did already on that. Let's look at the methylation one. So I'm going to run the next line for that one. So right, that's the one where it's either unmethylated or methylated, and there's some in between. You can see that there's definitely two peaks there, a low and a high, right? I mean, there's a big gap in the middle where hardly any values are right around 50. You have hardly any. And then the third one, the chromatin accessibility, will run this line as well. Same deal there. There's kind of a low half, a gap in the middle, and a high one, right? So yes, according to the author here, the first one, the RNA-seq, is kind of like, yes, it doesn't look like a perfect bell curve, but it's close enough to roughly approximate it with a normal distribution. But clearly the other two would be terrible approximation with a normal distribution, because they're clearly kind of like to a bimodal data set. So we can, what they recommend doing in that case is to pick a threshold, say everything below the threshold is a no or a 0, everything above it is a 1, like everything below threshold is unmethylated, everything above is methylated, right? And so we'll just convert those second two omics data sets into binary data. And since they range between 0 and 100, and there was a gap around 50, we'll use 50 as the cutoff. And so this line basically is telling us if it's less than 50, make it a 0. If it's greater than or equal to 50, make it a 1. And then look at the first few rows and columns to see what it looks like now. We're going to do that for the methylome data. If else SCBS seq is less than 50. If it's less than 50, it's going to be 0. If it's greater than 50, it's going to be 1. So hit Control-Enter to run that line. And look at the first few rows and columns with the next line. you can see now instead of 0, 100, occasionally 30, we have zeros and ones. And now, because oftentimes when you have binary data like this, we have less variance now, because like we could have had data that had some variance before, but now that column or that row of data is all zeros or all ones. So we're going to do that variance filtering step where we get rid of loci with little to no variation again. Now that we've turned it to binary, we're going to do that step again. So we're gonna do the near zero var again on this new binarized version of our methylome data. So I'm just going to run it one line at a time so you can see what's going on. First, we'll calculate the ones with low variation. Take a look. Okay, we still have quite a few that have low variation. How many? 700 more loci have been identified as having low variation than before. So the next line gets rid of those rows from the data. Take a look, right? Again, just take a quick look to make sure we didn't mess things up and delete all the data or something. Okay, the data are still there, but less. And how big is it now? We're down to 4,500. Where we started at 8,000, we went down to about 5,000. Now we're down to 4,500. And yeah, the next 7 or 8 lines do the exact same thing for the third omics data set, for the chromatin accessibility data set. We're going to run that again. 50 is the cutoff because it also was between 0 and 100. Run that. Okay. Do that line there. Take a look in line 82 to see there are some that we're getting rid of. How many are we getting rid of? Another 2,200-ish. Line 84 is the one that actually deletes those rows from the data. Let's take a look at the first few rows to make sure we didn't mess things up. Okay, so all looks good. And what do we have left? We're down to 9,000 and some. We've gotten rid of another decent number from our original 12,000. Okay. Any questions or anybody need help with anything? Awesome. So let's go back to the code. So that's what we all did. Okay. Now we've done all our data pre-processing that we need to do. Let's actually run the factor analysis on this to look at patterns across our 3 omics. So the next line of code basically takes the 3 omics and puts them together in a list of 3 objects. A list is just a data structure in R that can combine multiple objects into a list of objects, and then we can take a look at the dimensions of each of these 3 matrices that we have, make sure they're all match up. So that's what we're going to do. And so we want on line 90, we're going to make a list. And we're going to convert each of the objects into a matrix using the function as.matrix. A matrix is a more simplified version of a 2-dimensional data structure in R. A data frame can have all different data types. A matrix can only have one data type in it. So these are all numeric data. So we're converting it to a matrix because that's just what the package requires the data to be in matrix form. So we're going to hit Control Enter to run that and create a list called omics. And then this next line, lapply omics, comma dim, will give us the dimensions of each matrix in that list. Run that. So you can see they all 3 have different numbers of rows because there's different features in each of the omics data sets, but they all have 113 columns because we have the same samples. Those are 113 cells that 3 omics data sets were gathered from each of those cells. So then The next line is create MOFA from matrix, which basically takes that list of matrices and turns it into a special class that will work with our MOFA analysis package, multi-omics factor analysis. Hit Control Enter to run that. It's going to be called MOFA object. Okay. And then this function plot data overview can be used to see if there's any missing data. It just makes a quick plot. In the next example, it will have some missing data, and you'll see there'll be like grayed out areas where some data are missing. But here, this is just like clean example data that doesn't have any missing values. So it just kind of shows a solid color for each one, because no samples have missing data. But this is just a step you'd normally want to do to make sure everything is as it should be. Again, if there was some typo in your code, you might see gray there, and so that would be a sign you have to go back and check things. Plot underscore data underscore overview is the name of the function. Control Enter. Okay, there's the plot as it is in the notebook. Next step is we have to set some training parameters for the model, such as the distribution of the statistical distribution that each of the omics is following. And then there's also some other options like how many iterations a model will run for and how many factors we're going to compute. Now, I think the number of factors to compute is basically a function of how much computing time you want the model to run for. So you can kind of set it as high as you're comfortable with, since you're only going to be looking at the first few factors anyway. So this person set it to 20, which seems like a reasonable thing to set it to, because you sort of want to reduce the dimensionality. It doesn't make sense to do too many, because then you're not reducing the dimensionality that much. So we're going to get the default options, data options for the object, and then we're going to keep those all as is. We're not going to change them. Then there's three sets of options: the data options, the model options, and the training options. We're gonna keep all the defaults for the data options, but we're going to change some of the ones for the model options, right? Because remember, we said, okay, our RNA-seq is more or less normal or Gaussian, so we're going to call that Gaussian. And the other two, Bernoulli distribution is used for binary data that are zeros and ones. That's what we're going to use for the other two, the ones that are either methylated or unmethylated or accessible or inaccessible. We're going to use Bernoulli distribution for those. And then set 20. So then the likelihoods, those are the statistical distributions, are set to those names of the distributions. And we're going to compute 20 factors. Okay, so we're going to get default data options, run that line, take a look at them. So yeah, these are like, are we going to scale? Apparently by default it does not scale, but it does center. It subtracts the means off of them, which I guess is like an acceptable default. And then this line, get default model options, but we're going to change the distributions. The first one is going to be Gaussian. The second and third are going to be Bernoulli. And we are going to compute 20 factors. Run all that there. So there we have our distributions and our number of factors. Now, the next one is our training options. We're setting a random seed to make sure we all get the same results. This is kind of a important parameter, which I'm not exactly sure how the author decided that this was a valid one to use. Probably you would have to experiment in your actual work. You would have to set this threshold differently and see if it affected your results. Basically, it will determine how many factors we will keep in the end by getting rid of factors that explain less than 3% of the variation across all the omics. So like if it's explaining less than 3% of the variation is not a meaningful factor, and it'll not be retained in the final analysis or in the final results presentation. Maybe you want to reduce that threshold to keep more factors. But again, like if it's not explaining much of the variation, it doesn't really have a very good biological interpretation anyway. So apparently here it is thought that 3% is a good cutoff, but you could probably use other cutoffs depending on your research question. And 1,000 iterations, which I guess again is it does not get to 1,000 iterations. That's just to make sure that this thing doesn't run all day. It actually arrives at a solution before we get anywhere near 1,000. But that's just like something you set to make sure that the thing stops in a reasonable amount of time. And verbose equals true means it will print all the progress indicators as it goes. So the only thing you need to type in is 0.03 for that variance threshold. Control Enter to run all of that. Any questions? Cool. So let's see. Next step is we're now actually going to run MOFA. Now here's where I would say you have two options. If you really want to run this, you either have to have Python installed on your machine, or I've set this option use basilisk equals true, means the first time you ever run MOFA on your machine, it will on the fly kind of install a copy of Python that just has the packages you need to run this analysis. I mean, if you want to, you can run that. It'll take 5 minutes at least to run. So it's up to you if you want to do that. I've also just posted on the website our output for the model that's already been run, which you could just download and look at, like just for today's learning purposes. If you don't want to bother running everything and waiting for the thing to load and all that stuff, you can just skip the run MOFA line and just read our DS from the website. That would be probably the easiest thing, unless you if you already if you ever want to do this again though, you should go ahead and run it, and then and it will it only has to install everything the first time. So that's kind of up to you there. So we're going to use the prepare underscore MOFA function to set all the options that we just went through and identified one by one. I'm gonna run this line. Okay, and now it gives us a warning that some views have a lot of features. It is recommended to perform a more stringent feature selection. So right, even though we did filter out a lot of features that had low variance, it's telling us there's still kind of a lot, but we're just going to go with it anyway. Again, you could have set the threshold for low variance a little bit higher or something like that and gotten rid of more if you wanted to. Okay, so I'm just going to go ahead and run this just to show you how long it takes to run once you already have everything. Once Python is installed, it doesn't take too long to run this model. Yep, so we'll just let it run, basically once it is run, what we're going to do is spend the rest of this demo kind of like looking at the results from it, and making some graphs and trying to explain patterns of variation in the data based on what we see, like coming up with biological interpretations for the factors that MOFA has identified as being important across all the omics. Yep. So basically it's you can see it's now running. It is going through multiple iterations and giving us a little progress indicator each time. Should only take a few more seconds to finish running. There we go. So it takes, you know, after it gets initialized, it only takes a minute or so to run. And yes, we get a warning message that it saved everything to a temporary file, which is fine. And the other warning message is that factor 1 is strongly correlated with the total number of expressed features for at least one of your omics. Such factors appear when there are differences in the total levels between your samples, sometimes because of poor normalization in the pre-processing steps. So we didn't normalize for particular cells having higher overall expression than others, which might be sort of a confounding variable in our results. So that would be yeah, there are kind of like other transformations we could have done on the data that may have controlled for that better. But let's just kind of not worry about that for today. Okay, so yeah, so anybody who doesn't want to go through the steps of actually running it can just run line 130, skipping the run MOFA and all that, and just run line 130, and it'll give you the exact same thing that I have without having to actually fit the model on your own laptop. If I just hit Control Enter on this line 132, it'll print some basic information about the model we just ran. So here we have it. It just tells us like the number of views, in other words, the number of omics data sets that went into it, how many variables each one had, and how many samples there were. And right, since we are doing a sort of unsupervised analysis, we didn't tell the model which groups each sample belonged to, so we just put everything in one group. And oh, this is important, it only kept 3 factors. So out of those 20 that it computed, only 3 of them explained at least 3% of the variation across all the samples. So we're only going to be looking at 3 dimensions in our data, 3 dimensions. Is everybody with me? Is anybody unable to get this so far? Or are we all good? Cool. Okay, so let's see, back to the looking at the output. Basically, what we're going to do first is look at how much variance was explained by each of the 3 factors that our factor analysis identified as being explaining a decent amount of variation across all the omics. First of all, we're going to look at this sort of R squared of each of the 3 omics. How much variation across the 3 omics is explained by by the 3 factors combined, and then looking at it separately by factor as well. So we're going to do that. The function that pulls out that variance explained from our fitted model object is get underscore variance underscore explained. And once it starts typing it, I can hit tab to complete it. I'm going to hit Control Enter to run this line 137 right here. So here we can see that our 3 factors explained 13% of the variation in the RNA-seq and about 5% of the variation in the other 2. So it's not a huge amount, but you know, I don't really have a way to contextualize that number, but it's you know, it's 13%, I guess, is a decent amount. And then line 138 breaks that down by the 3 factors. So yeah, so this the 13, right, can be broken down into about 7% for factor 1, 3% each for factors 2 and 3. And then if we run this get variance explained of of the MOFA object, it'll give us the basically the same thing. Now, we can also, there are some functions in the MOFA2 package that allow us to make some plots that will show this variance explained stuff graphically. So let's do that. Yep, we're going to use the function plot variance explained. X equals view, that means the x-axis is going to be each of the omics, and y equals factor. Means plot the variance explained by each factor, and we're going to look at the total. And so here's the, you can make a bar plot of the variance explained, which is the same numbers as we just saw on the 13, 5, and 5. You can just make it in a plot form. So that's that line one for the plot underscore variance underscore explained. Control Enter. Now I'm going to run line 144. That's going to pull out the second element of this plot list we just made. That shows the total variance, the 13, 5, and 5. And if I hit Control Enter on the next line, it's going to show it separately for each of the 3 factors as kind of like a heat map. So what do we see here? Well, here we see that there's one factor, factor 1, that explains a decent amount of variation across all 3 of the omics. But factors 2 and 3 only explain a kind of non-zero amount of variance in the RNA-seq. There's the other 2, methylation and chromatin accessibility. Those, for factors 2 and 3, their variance explained is close to 0. So there's really only one dimension, one factor in the data, factor 1, that has explanatory power across all 3 of the omics. That's what this is showing us. So now let's say, okay, well, that's great that we found these factors, but what do the factors even mean, right? Those are just kind of like parameters in a model. They don't really have any intrinsic biological interpretation until we go back to the omics and look at which genes, which loci from our data set are associated with each of those factors. And then once we look at those, we can kind of try to come up with a biological interpretation for what those factors even mean. And this code here, get factors, pulls out each of the factors from our object, and then we can plot weights, which means plot the how much each of the genes in our gene expression data set are associated with factor 1, the factor from our analysis that explained the most variation across all 3 of the omics. Plot weights will do that. Okay, so we're going to use get factors, get underscore factors to pull those out. That's just telling us there are three factors. And then we're going to use plot underscore weights. Let's see, what's it? Yes, view is the term they use for which omics it is, and factor equals 1. View is the RNA-seq data set, and the single cell RNA-seq, and factor equals 1. Run that line. Okay, so basically we have, what this shows is that each point along this like S-shaped curve represents one of the genes. Many of them are near 0. Many of them have no association with the factor 1, but a few that are kind of highlighted here have a negative association, and some have a positive association with it. So these are like cells that have a high value of factor 1 have a high expression of these genes. Cells that have a low value of factor 1 have a high expression of these genes. That's what, so like if you knew anything about these genes, you might be able to come up with a biological interpretation for that. Any questions? Okay, and then this is just another, there's some other functions in the MOFA2 package that just give us like some different ways to plot the most highly positively and negatively associated genes with each factor. And you can, like, if you want, you can change up the numbers in this argument, like you'd look at factor 2 and things like that. Plot top weights basically just isolates those genes that have the highest positive and negative associations with the factor. We're going to look at the RNA-seq again. We're going to look at factor 1 because that's the one that explains the most variation, and we're going to look at the top 10, whether positive or negative. And then we'll be able to generate this plot right here. Plot underscore top underscore weights. Yeah, in the example, they're plotting the top 10. Obviously, you could plot a different number if you wanted to. Hit Control Enter there. And so, yeah, this just shows like, okay, this gene HPRT is highly positively associated with factor 1. FAM221B has a negative association. But these 10 genes have the strongest association across all the thousands in the data set. And you can also plot a heat map with looking at the top 10. Again, we're looking at the same top 10 across the thousands in the data set that have the 10 strongest associations with factor 1. And it'll tell us what is their expression level in each of the cells. So let's take a look at that. And so, yeah, ones with a high expression will be sort of red, and ones with low expression will be blue. Plot underscore data underscore heat map. Run that. So let me zoom in on this so we can have a closer look. If you can see this, you can see that a couple of, right, these were the genes, the ones that are, the 8 of them that have a lot of red and orange on the left side are the ones that have a high expression in high values of factor 1. There's a couple that have a more expression on the right side. Those are the ones that had high expression with low values of factor 1. And now if you look at it, look at the names of the samples, you can see that these ones are all ESC and these ones are all EB. If you remember, I told you at the beginning there were 2 different cell types, the embryonic stem cells and the embryonic bodies. Well, it looks like you can see like right where this cutoff is the line between the embryonic stem cell samples and the embryonic body samples. So to me, it seems like factor 1 is basically a factor associated with whether the sample was taken from the ESC cell lines, cell type, or the EB cell type. So this, like, well, it looks like certain genes have a higher level of expression in stem cells, and these ones have a higher expression not in the stem cells. And so even though it was an unsupervised method and we didn't tell the model which samples were stem cells and which were not, it seems to have pulled out an axis of variation that is explaining a lot of overall variance in the data that's associated with whether the cells were stem cells or not, or embryonic bodies, right? Does that make sense? Okay. What can we do next? We can do the same exact thing as we did for the RNA-seq, except we can do it for the bisulfite sequencing, the methylation data set in factor 1. So this is just like changing the view argument to the BS instead of RNA-seq. So I'm just going to run these lines of code here real quick, so because it's the same exact thing, except we're doing it with the other 2 omics. You can see, okay, here's a few genes that are highly methylated in high values of factor 1, and here's some genes that are highly unmethylated or some loci that are highly unmethylated in high values of factor 1. Line 156 just shows that in a different way by looking at these weights. And it makes sense that there's a lot more low methylation in the stem cells because that's where a lot of biological activity is going on and a lot of genes are being expressed, so they're not methylated, right? Okay, so let's look at this heat map again for that one on line 157. Yep, so you can see we have, right, because the data were zeros and ones, there's only two colors on it. One is the I guess the unmethylated, and 0 is methylated. And so we have again, there looks like there's kind of this binary split between the embryonic stem cells on the left and the embryonic bodies on the right. You can zoom in and take a closer look if you want. And we'll do the same thing for the chromatin accessibility data set on the next 3 lines. Here's the weight plot. So all the top 10 are on one side there. There's the top 10. Again, they're all positive associations with factor 1, which I guess refers to higher accessibility. And or I'm not sure exactly, but then here's the you can see there's kind of this binary cut off between the stem cells and the embryonic bodies again. Everyone good. So we only have a couple of lines left in this first demo, and then we'll take a little break. So that was that. We just went through all that. Now the last thing that that we can do is so the conclusion that is drawn from this is, okay, the features that contributed the most variation to factor 1 may be correlated with each other because they're involved in the same biological pathway. So they would be, right, this doesn't really tell us that, but it gives us information to form a hypothesis about the fact that those features, those genes might be involved in the same biological pathway. If you actually wanted to test that hypothesis, you would have to do additional studies targeting those features that we've identified. So this is like, it's just part of the scientific method, right? It's just kind of the exploring part, looking for information and patterns that will be used to form a hypothesis that we can later test with other studies. Because scientific process never ends, right? It's a cycle. Okay, so the last thing that that we're doing with this example is to as I mentioned in the introductory slides, a lot of times people will do one of these dimension reduction methods and then use a machine learning based algorithm to kind of like further reduce the dimensions and visualize the results to just plot how these all shake out. And it's kind of just like a way to make pretty pictures showing that there's separation between the groups in your data. Here, as it turns out, some of the cells were control cells, and so this line of code just identifies which of our 113 samples were controls, and then which is going to be used for plotting the data. So we're going to, yes, we're going to set the column names to control cell if the sample name appears in this list of control cells we've identified. So we're going to change the column names of our RNA-seq data if they appear in the controls vector. We're going to change those column names to control cell. So we're going to run these lines of code to do that. Then we're going to, since we didn't, remember we never told the model which cell types the samples belong to. Now we're going to actually add that metadata in just for plotting purposes. And so we're going to make a data frame of sample metadata saying which cell type, stem cell, embryonic body, or control cell each of the samples belong to. and then we're going to assign that object to the sample metadata of our object, of our fitted model object. So this is called samples underscore metadata is the function that we use to access or change the metadata of a pre-fitted object. So we'll run these 3 lines to do that. So yes, it's telling us, you know, this sample comes from an embryonic stem cell and so forth. Everyone good? Yeah, we're about done with this first demo here. Basically, the next few lines are just running 2 different forms of machine learning based dimension reduction to visualize how well our factor analysis was able to distinguish between the different cell types. And it just makes a nice way to plot that in 2-dimensional space. And we're going to use two methods for that, UMAP and TSNE, which are just 2 different algorithms for doing that. And each of them has, right, sort of like a regularization parameter because they're machine learning methods, as we learned yesterday. And we're just going to set them to the square root of the number of cell types, basically. And perplexity, number of neighbors is the one we use for the UMAP, and perplexity is the name of the regularization parameter for the other machine learning method. So run underscore UMAP is the one we're going to use for the first machine learning method we're going to use for visualization. And number of neighbors is the regularization parameter for that. And run underscore TSNE is the other one, and perplexity is the name of the regularization parameter. And we're going to set a random seed so that you all will get the same results as I do on your screen. Let's run that. It should just take a few seconds. Okay. And then, oh yeah, then we're going to use plot dim red, plot dimension reduction. And we can color it by cell type or condition. We'll plot the, this is the TSNE for it, and just making slightly bigger points so you can see how things shake out. Plot underscore dim red will plot the dimension reduction. Which method we'll use, so we can just see how well each of these 2 machine learning methods did at visualizing the separation between our cell types. Color by condition, which is the name of the column containing the cell type. We'll hit Control Enter on this 178 to run that. So yeah, you can see that it did a pretty good job. There's like a very wide separation between the cell types. One of the control cells kind of got included in with the embryonic body cells, but other than that, there's like pretty much perfect separation between the groups for this method TSNE. We'll see how the other one did, UMAP. Run that line. So yeah, same deal. Basically, it apparently one control cell got misclassified again by this one, but other than that, they're very well separated. And we can basically plot those same dimension reduction plots colored by any of the 3 factors in our analysis to take a look at how well those factors correlate with the reduced dimensionality identified by the machine learning methods. So we'll do that for each factor. We'll take a look at factor 1. Hit Control Enter. Yeah, here we see that these ones, I think this one on the left, those were the embryonic stem cells, I think, if we look back. Yeah, they were the embryonic stem cells, have a high value for factor 1. The embryonic bodies have a little bit lower, and the control cells have a very low value for factor 1. Let's look at the same with factor 2. Again, factor 2, yeah, it looks like factor 2 is doing a pretty good job separating the control cells from the other two. Those have a high value of factor 2. And lastly, factor 3. It looks like factor 3 is not really associated with variation between the groups, right? Because we have kind of high and low values of factor 3 in all three of our clusters. So factor 3, whatever biological interpretation it has, is not associated with systematic differences between the cell types. Okay, well, I've been kind of talking at you all for quite some time here. That's kind of the end of the first demo. Does anyone have any questions about this here? Okay, well, I guess for people in Central Time, it's still a little early. So why don't we, let's take like a 10-minute break, do another little bit, and then take a longer break for lunch. I don't think we'll get through all 5 of the demos today, but that's okay because I kind of just provided a lot of material just so we would have plenty to get through. And I don't want to like overdo it because I want people to have, you know, not hate me and hate themselves by the end of this thing. So let's take a 10-minute break, come back at 11:50, do a little bit more, take a lunch break, and then after lunch we'll come back, maybe do another 1 or 2 of them, and then I'll either have like office hours Q&A or people can just peace out. So let's take a break until 11:50 Central Time.
Dr. Reed, it looked like
Yeah, what's
maybe
up?
the RDS link wasn't working.
Which one?
The last one. I know you can also get them from that zip file, but I think
The single cell one did not work?
yeah,
This one here?
that one. Yeah.
It didn't? Oh, shoot. Let's see.
And I tried some of the on the next demo, a couple of them didn't work too. So
Did I?
not sure.
Okay, let me okay, cool. Well, you should have said something about that sooner. Well, I will look into why that is, but wait, so they didn't work on the next demo either?
One of the I just tried one of them on the next demo. It didn't work either. Yeah.
Okay. Yes. Well, thanks for letting me know. Let me just let me try to figure that out real quick.
Okay.
Alrighty. Well, I think first of all, I said in the chat a second ago, I apologize for any issues people had importing the object that was the model object. I figured out a solution for it, but I'm pretty sure that we will probably I think what I'm going to do is I'm going to go ahead and go to demo number 3, because I think demo 2 is just a little bit more in-depth version of the same thing we just did, which you're free to look at on your own time. But I think if we're not going to do all of them, I think it would be more interesting for you all to have exposure to more variety of methods. So I'm going to before we have a longer break, I'm going to do the third of the 5 demos, which is looking at the supervised method, partial least squares analysis to look at a different example data set. So I'm going to go ahead and go back to screen share here. Okay, so I'm back on the course web page, and I'm going to open up the course materials for the third of the 5 demos. We're just going to skip number 2 for now. Okay, so basically this is a, let me make it a little bigger here. We're going to use the MultiOmics package to do a sparse partial least squares discriminant analysis. This is not MultiOmics. We're just looking at one omics data set taken from different types of tumor cells, and to look at whether there are signatures in that I believe it's a gene expression data set that will allow us to predict what kind of tumor we have, basically, whether the omics differ between the tumor types in a way that is predictive. And this was from a paper published in 2001. We have about 2,000 genes, so a not too huge data set, and there were 63 tumors that they sampled for the expression of these genes. And there was 4 types of tumors. You can see a lymphoma, sarcoma, and so forth. There's 4 types. So we have 63 samples. Each sample has expression levels for about 2,000 genes, and each sample belongs to one of 4 tumor classes. And these are the so there's an example data set that comes with the MultiOmics package. So we're not going to read it in from a CSV file. As long as you have a MultiOmics package installed, you have this example data set on your computer already. Right. So this is a supervised analysis because we know what group each one belongs to, each tumor. We know which type it is, and we're going to incorporate that information into our statistical model. So there's the SRBCT, small round blue cell tumors, is a list with 3 elements. The element called gene is the matrix of 63 rows samples by 2,300 columns, the genes. Class is just a vector that's 63 elements long, which is a factor that has the tumor type for each individual. And gene name, it gives us more textual information about the genes. Okay, so as I told you in the introductory slides, it's a good idea to do a quick PCA on your data as a preliminary investigation to just get to know the structure of the data before you use that old tried and true method, before you go and do fancy multivariate methods on it. So we're gonna so what I would recommend doing is restarting your R session and just clearing everything so you have a fresh session and you don't accidentally have any variables cluttering things up that will cause you to get different outputs than I do. So we're gonna once I go back and open the worksheet. I'm going to load the MultiOmics package, and then I'm going to load the example data set using the data function, which is used to import example data sets. I'm going to assign the gene object from that data set to a variable called X. I'm going to look at how big it is, and I'm going to do a quick PCA on a principal component analysis, the simplest and most old and reliable multivariate analysis on that data set. Okay, we're going to do that real quick. I'm going to go back to RStudio. I'll clear away everything from before. I'll even restart the R session I have here, so everything goes away and everything's nice and clean. And now I'm going to open the worksheet for demo 3. Okay, I'll load the MultiOmics package by hitting Control Enter on there. And then I'm going to import the SRBCT, small round blue cell tumor data set. I'll run that line. I'm going to run the next line to create a variable called X, which is the sample by gene matrix. I'm going to use dim of X to look at how big that is. It has 63 rows, one for each tumor, and 2,300 columns, one for each gene expression in the data set. Now I'm going to run a PCA on it. And I will, in the example, they use 10 principal components, but doesn't really matter how many you put as long as you put enough to see where most of the variation is. Center and scale means we're standardizing all the variables before we do the PCA, which is good to do in multivariate analyses. I'll run that, Control Enter. I'm going to print information about the PCA. You can see that here, this is the proportion of variation explained by each of the first 10 principal components. So one axis of variation explains about 11% of the variation across all 2,000 genes. The next explains another 11%-ish. Then we go have 6. It always in decreasing order, 5 and 4, and so forth. And if you make a plot, it will give you a bar plot of those proportion variants explained as well. And that's the same numbers as we just saw. So this actually has some like sort of interactive questions. I don't know if you all are feeling the interactive vibe right now, so I won't force you to answer. But because we're going to go through these answers as we go. But basically, how many principal components would you choose to capture most of the variation in this data set? Well, you could say, well, we definitely don't want to leave out components 1 and 2. They explain a lot of variation, but there's clearly like a drop off between 2 and 3. Maybe I would include like 3 or 4, but yeah, definitely at least 2 would be pretty informative for explaining. There's clearly a gap between 2 and 3 there. So there's a function called plot indiv in the MultiOmics package that will let us look at how well, right, because PCA is an unsupervised method. We did not tell PCA which of the 4 tumor types those samples belong to. We only gave it a bunch of gene expression data and said, okay, tell me where the variation in this data is. Let's see if we then look at how separated those tumor types are by the PCA, that's just going to tell us, well, is there sort of natural separation between the 4 tumor types that PCA was able to pick up, even though it didn't know which tumor type each sample came from. So we're just gonna plot the 2, because right, PCA axis 1 and 2 explained a lot of variation in the data. Let's plot each each of our samples data points in the first two axes of PCA space and color it in by the tumor type to see if they're separated out. Okay? That's what we're going to do here. Yes, I don't know where this ELT came from, but you want to change that to plot in div. Okay? That's the function that plots the individual data points for in PCA space. We're going to use components 1 and 2. And the group we're going to color it by is the class column from the SRBCT data set. And this will give us it colored by their cell type to see how well the PCA did. Control Enter on this. Okay, so we can see here that it didn't do, at least the first two axes did not do an amazing job of separating out the tumor types, right? Like there's like it kind of did, right? Because the BL cell type always has kind of a low value of PC1, the triangles over here. You know, this area only has EWS and RMS cell types, but they're not very neatly separated. So there's some variation within the cell types as well that is impeding our ability to tell apart the cell types just from these two axes, right? There's also, if you want, you can make a 3D plot to show the PCA in components 1, 2, and 3 all on the same plot, which you know, is just more of a gimmick, but I'll show you anyway, just in case you are interested in that. It should pop up in a separate window. Yep. So you can see it in 3-dimensional space. Once again, it can be seen that there's some, you know, it's doing some separation, like the red and green cell types are in one area and the blue and black in the other. So it's kind of separating one group of 2 from the other group of 2, but it's not perfectly separating the 4 cell types, even with 3 dimensions in PCA space. Any questions so far? Okay, so let's see. What will we do next? Well, we're going to next make a correlation circle plot, which will show us for each gene how highly correlated they each are with each of the first 2 PCA axes. And you can see that the 2 axes on the correlate, well, I'll show you it using the plot var function, which will show us that plot. Hit Control Enter. So you can see there's like, it looks like a bullseye with a bunch of bullet holes in it. Basically, anything that's near the outside of the circle is highly correlated with one or more of the PCA axes, like it's near +1 or is near -1. It's highly correlated with axis 1. The ones way up here and way down here are highly correlated with axis 2. Everything inside near the middle, those genes are not very correlated with the axes that are explaining most of the variation overall. So these ones are probably less informative for us. The ones on the outside are important. Now, okay, well, that was kind of good for starters, but let's do a couple of analyses, partial least squares discriminant analysis and sparse partial least squares discriminant analysis to see if we can make a predictive model using the groups we know these things belong to predict new samples. So what we have to do is, well, we're going to create objects called X and Y. X is the matrix and Y is the X is the matrix and Y is the vector of tumor types. And then we're going to use summary to look at a table of how many belong to each type, and then look at the dimensions of the matrix and vector. So X, we're going to create a variable called X from the gene object in our example data, and a variable called Y from the class object in our example data. We're going to hit Control Enter to run this line, run this line. Now summary of Y will give us this telling us how many of each of the 63 samples come from each tumor type. Dimensions of X, 63 by 2000, and the length of Y is 63. So that's good because we want to have gene expression data and tumor class data value for all our samples. So we have the same X and Y for every sample. So the first thing we're going to do is we're going to do a partial least square discriminant analysis. This is not the sparse one yet. This is not doing any variable selection. It's not doing any regularization. It's actually using all the variables in the data as much as possible and not narrowing it down. And it has the potential to overfit to the data. It's not doing that machine learning style regularization yet. But we're going to start with PLS before we go to sparse PLS. So we use the function PLSDA to do this, and we're going to use 3 components. Because it is, and remember, this is now a supervised method. We are telling it which group the samples belong to. We are not only looking at finding axes that explain as much variation in the gene's expression as possible, but we're also looking at axes that explain variation in gene expression associated with what groups they belong to. We're actually using that grouping variable in the model parameter estimation. And then we're going to plot axes 1 and 2 of that. We're going to use the PLSDA function, and we're going to use 3 components. N comp, number of components, equals 3. We're going to run that line. And then we're going to make a plot of axes 1 and 2 of SRBCT.PLSDA. And we'll plot a legend so that it will color it in by the group names. Okay, what do we see here? We see, obviously, this is separating things a lot more cleanly than the PCA, right? Like all the ones of each tumor type are in their own little group, and there's none of them are in each other's group, and there's a big gap between each of the 4 groups, right? But it's like, well, of course it did, because we told the thing what groups everything belongs to, so it was able to find genes that were associated with the differences between those 4 groups and pull them out. But maybe it's kind of overfitting to noise. Maybe there's just random noise in our data that happens to be correlated with those groups, and it's overfitting. How do we know we didn't just overfit? Well, we have to use a machine learning approach where we do the same thing except with regularization to keep us from overfitting to the data, and then cross-validation to predict on data that were not used to fit the model. And if it does good with that, that means we have found signal in the data that's actually real and is associated with real differences between the groups and is not just random noise, right? That's what our goal is now. That's what the sparse PLSDA is going to do. Now, we will, the way that sparse PLSDA works is that we select a number of components to estimate and how many variables will be retained on each component. The non-sparse PLS retains all variables for every component. Sparse PLS, the way it regularizes and avoids overfitting is by turning a lot of variables association with each component to 0, so the model has fewer parameters. And later, we're actually going to use a cross-validation to figure out an optimal number of components and an optimal number of variables per component. But first, just to show you how it works, we're going to just do it with preset numbers for each of those things. We're going to use 50 variables on component 1, 40, by variables I mean genes, 50 genes associated with component 1, 40 on component 2, and 30 on component 3, and select 3 components. Again, those are kind of picked almost arbitrary later, and in reality, we would use cross-validation and test many different numbers for each of those things to determine what is ideal. But right now, we're just doing it real quick to demonstrate. So again, this is like the PLSDA we just did, except we're purposefully reducing the number of variables we're allowed to use to explain the variations so that it doesn't overfit to the data too much. Okay, so this is our first sparse PLS. We decided to use 50 on component 1, 40 on component 2, and 30 on component 3. And so it has, yes, the function is SPLSDA. NComp, number of components is still 3, but now this new set of parameters that the regular PLS doesn't have, but sparse PLS does have, is keep X. How many variables are going to be non-zero loadings on each component? Keep X is that. We're going to run line 45 to set that vector of 50, 40, 30, and then we're going to run the sparse PLS on line 46. It's very quick to run. It just, with our small data set, and it's a simple algorithm, it ran in basically no time. So let's take a look at our plot the individuals for that SPLSDA.SRBCT. How does it look compared to, like, well, look at this before it goes away. Take a look at the results we had for PLS, right? It had a very clean separation. How's it going to look when we took away some of our ability to predict by only allowing ourselves to use a few variables at a time? Let's look. Like, okay, well, what's the difference here? Well, you can see that it still, with 2 components, is able to separate the BL from everything else, and it's able to separate EWS from everything else. But 2 components, now that we've reduced our sort of how closely we fit the data, are no longer enough to separate these 2 tumor types from each other, NB and RMS, right? It doesn't separate them out as well. So what do we do? Oh, and well, but before we do that, let's also take a look at the correlation circle plot. Remember that with the PACA and with the PLSDA, we kept all variables in, regardless of how highly correlated they were with our axes. Sparse PLS is keeping only the ones that are most explanatory, that are most correlated with our axes. So you can actually see that the correlation circle plot is going to reflect that. So we're going to use that plot var again, and this time we're actually going to plot the names of the genes on the plot as well. And we're going to use components 1 and 2, C1, comma 2. Control Enter. So now you see the correlation circle plot only contains genes that are in this outside, like, donut area, which has a high correlation with one or more of the PCA axes, right? Yeah, I think let me we're not going to get done with this one before it's like a good time for lunch. So I'm gonna do a couple more slides, or go through a couple more things, and then we'll take a lunch break. Since I know you all are probably hungry for lunch, and I mean I am too, because it's an hour later here anyway. So we'll let me just do a couple more, and then we'll take a solid break. Since well, we'll just get to a good stopping place here somewhere. Okay, so yeah, the next little bit here, basically what the next part of this demo is just doing some more diagnostic plots to look at the output of the sparse PLS. Then we're going to actually do, since this is just like one that we picked the number of variables kind of out of thin air, we should really do cross-validation to do that. So we're going to do that next, and then we're going to assess the model performance after that. That's what the rest of this demo is about. So plot loadings is another diagnostic plot that you can use to show the output of your sparse PLS. So the first argument is the sparse PLS object. Which component are we plotting? And these are sort of like, there are different ways to rank how strongly each one contributed. We're going to use the mean method and which ones had the highest contribution. And so we'll take a look at what this shows. Plot loadings is the name of the function. And we're going to do component 2, just as an example. And hit Control Enter. And here you can see these are the names of the genes. Again, they don't really, you would have to look up what genes these are and what potential biological function they have. Let me just zoom this in a bit so you can see the whole thing. So you can see that some genes that have a very low level of expression are contributing to distinguishing the EWS gene cell tumor type from the other ones on component 2, and then you can interpret the other ones similarly. Yeah, and let's change component 2 to component 1 here, just to take a look at which cell type, tumor type is being separated out by component 1, because it seems like component 2 is mostly being used to distinguish EWS from the other ones. Component 1, yeah, so component 1 you can see is all orange, right? Component 1, apparently the BL tumor type is very elevated in expression of a lot of genes that the other 3 cell tumor types do not have very high expression in. So it's, and that's a very strong signal, right? It's the axis explaining most of the variation. So the most kind of strong signal in the data is that this BL, whatever lymphoma tumor we're dealing with here, is highly elevated in expression of a bunch of genes that are not very highly expressed in any other type of cancer tumor that we have. That's what this is showing us. Okay, I'll do a couple. I'll just do the last few slides or lines of code dealing with this first sparse PLS, and then when we come back from lunch, we'll finish this demo with our cross-validation stuff. So before we do cross-validation to determine the optimal number of parameters, what we're going to do is we're going to look at how good of predictive performance this sparse PLS DA does at correctly classifying tumor cells into groups based on their gene expression. So we're actually already doing cross-validation for this, like fitting our same model again to some of the data and using it to predict the rest without having basically using that portion of the data that was not used to fit the model predicted by the rest of it to see how well it does on data it hasn't seen yet. Again, this is not perfect because it's not we didn't go out and collect new data that are totally unused to fit the model. It's just sort of more of an internal validation. It wouldn't, if you really want to see how well this model does in the real world, you would need to get a whole new data set instead of splitting within this data set, because who knows, they're not, there might be some non-independence and you can never really avoid data leakage, correlation between training and test set in the internal split like this. We're going to use, we're going to set the random seed so that we all get the same results. Although if you change the random seed, you would get a slightly different result. 5-fold cross-validation repeated 10 times. So basically we're fitting this model 50 times real quick. So but since it is very quick to fit, that shouldn't take too long. M-fold means cross-validation here. And then we're going to look at the error rate for from the cross-validation. So perf is the function meaning assess model performance. And the folds equals 5. We're going to do 5-fold cross-validation repeated 10 times. That's a pretty reasonable number of folds and repetitions. So 5 and 10. So I'm gonna run this line where I set the random seed and then do the cross-validation. And it takes a few seconds because it's fitting everything 50 times. There we go. And now we can then print the error rate overall. So what percent of the time was it wrong when it was predicting the data points that were not used to fit the model? How often was it wrong? And you can see that there's actually 3 different distance metrics that we can use to, because the PLSDA gives us a continuous score, and then we can use 3 different distance metrics as cutoffs to decide which of the 4 classes it belongs to and see which one has the best performance. And you can see that it also gives us an error rate for each component separately. So if we only use one component, we are wrong about 43% of the time. And different distance metrics have slightly different performance there. If we use 2 components, we start to do a little better, 19% error rate there. By the time we use 3 components, we are only wrong about 1% of the time. And this is the overall error rate. There's also BER, which is balanced error rate. That accounts for the fact that we have unequal numbers of tumors in each of the classes, right? Like if 99% of the tumors were one kind and 1% was the other kind, we could always predict the most common one. We would not be wrong very often, but it would not be very useful, right? It would not be a useful model, but we would be right most of the time. But we can account for that imbalance and give us a balanced error rate, which are slightly different. I mean, our data are not too imbalanced, right? Because we had like 10 of one tumor type, 20 of another. They're not too imbalanced, so the balanced error rate isn't too different. It's not too different from our overall raw error rate. But if the data were highly imbalanced, those 2 numbers could be very different. And we can also look at the error rate by class. So this is our overall, but we could also say, well, how well did it do at predicting, or how often wrong was it for like the lymphoma one or the sarcoma or whatever. We can then see that for each class separately. You can see here the error rate for each one by the 3 distance metrics. You can see that if you only use one component, it was wrong 100% of the time about this NB tumor type. So like apparently component 1 does not convey much information about NB, but by the time we get to component 3, we have very low error rate for each class overall, or for all of them. Let's see. So lastly, before we do the other cross-validation thing, so this is the last thing I'll do before we take a break. You can also look at, this is kind of like a check on the robustness of our analysis. We can look at how often each variable, each gene is selected, right? Because sparse PLS only selects a few of the variables. If we do cross-validation, we're basically fitting the model to a different subset of the data many times. We can actually look at in each subset, was it selecting the same variables every time, or was it selecting different ones? If it's doing the same ones every time, that's a good sign because it means those variables truly convey a signal. If depending on what random subset of the data you pick, it's choosing different variables, that probably means it's kind of just like looking at random noise in the data. So we want our feature selection, which ones are picked, to be highly stable across the cross-validation folds. So the features that are the most frequently selected across the folds are the ones that probably have the most information, actual signal in them, and not noise. This is what those plots are going to show us. So I won't have you fill in the blanks there. I'll just you can just run this code here. Take a look. We can see that there's kind of like a lot of variation there. Some of the variables, some of the genes were selected across over 50% of the folds, and some were not. So this is just a plot of it, but you would want to go in and look in your in the output for which genes are selected most frequently, and those would indicate ones that are probably the most informative. Just another way of showing that. And you can also pull out the variables that were selected for each component and how strongly loaded they were on that component, and take a look at which ones have the highest loadings are not necessarily the ones that are most frequently selected. If one has a high loading and is most frequently selected, that's kind of a double indication that it is an important one, but they don't always match up. We can look at the run this for component 2. These are the genes with the highest positive or negative association with component 2. And then we could look at, if we run this, this is telling us which ones have both high frequently selected and high loading on component 2. So yeah, these ones, as you can see, the ones with the highest loading were selected 40% of the time, which is like pretty high on there, but there were some that were selected 60 up to 60% of the time, but those were not the ones with the highest loading, as it turns out, which is somewhat interesting. And so you might want to look at reasons for that discrepancy. Okay, well, I think again I've said enough here. What I would like to propose is to take an hour's break, and we can let's see. So it's like 12:30 where you all are. So we'll take an hour break, then we'll kind of like come back, do whatever we can in an additional hour, whatever people kind of want to do, or if people don't have interest anymore, we can either adjourn after that, or I will stay around for any Q&A, office hours, and anything like that. Oh, let me before we break for lunch, let me just show you one thing in case people don't. I mean, I will not be offended if you don't come back after lunch, but if you, whether or not you do, I do want to show you where the survey, post-workshop survey is, so you can fill that out. Yeah, so if you go to the course webpage and scroll all the way down to the bottom, there's a feedback survey here. It's very short and it's anonymous, and I would greatly appreciate it, even if, yeah, regardless of how much of it you participated in, if you could fill that out. Again, is anonymous. I would highly appreciate it. Does anyone have any questions before we take a break? Okay, well, let's take a. I'll just go ahead and I'll stop the recording as well, so we don't have a lot of dead time on there.